Well, a warm welcome to today's talk. It would be boring if I was on my own, so thank you for coming back. Now, yesterday we looked at pretty good news all the way through from the Oxford AstraZeneca vaccine in terms of preventing disease, in terms of preventing contagion, in terms of preventing severe disease. Well, today I want to look at the Sputnik V, the Sputnik vaccine, the Russian vaccine. And it's just published phase 3 interim results in the Lancet, fully peer-reviewed, and it looks really good. And we're going to look at it in some detail today. So I'm aware today's video is a bit geeky for some, so I'm just going to give you the headlines of it now. So it's interim phase 3 trials, peer-reviewed in the Lancet. The efficacy of the vaccine in preventing someone from contracting COVID-19 is 91.6%. Now, this is absolutely excellent. Very high efficacy in preventing disease. And after people have completed the course of the vaccine, the two doses of the vaccine, the first and the second dose, none of them were hospitalised. So 100% protection so far on the data we have against severe, well, actually it was moderate and severe COVID. So it's stopping people getting very sick 100%. So this is exactly what we want. It's looking really good. Now, safety profile also looks good. No adverse events or deaths were attributed to the vaccine. Now, we don't know, with the Oxford vaccine, we know that prevents cross-infection. With the Sputnik vaccine, Sputnik V, we don't yet know that. But I would be very surprised if it didn't. So, having had a bit of a evidence gap for the Sputnik vaccine, it's now completed and it's looking good. So to give you the complete bottom line on this video, would I have the Sputnik vaccine now personally? Well, based on the phase 3 data I have here, I think it very likely that a national agency would authorise this vaccine for use. And if my national agency authorised it, I would certainly have it. And based on the data we're going to look at today, I see no reason why they wouldn't authorise it. So pretty good news, and that's the bottom line. So if you want to skip it now, you can do, but if you want the details, let's go on to it. I'm going to start off with a bit of background first of all on this vaccine. Now, this is it here. So it's the Sputnik Gam-Covid-Vac is the name of the vaccine. 2 doses, 3 weeks apart, intramuscular injection, freezer storage. Now, there's 2 forms of this vaccine actually. The trial was actually done on one that has to be stored at freezer temperatures. It's not mega low temperatures like the Pfizer. It's about minus 20 normal freezer temperatures. But there's also a freeze-dried preparation that will probably be the one that's used for more bigger rollout programs if this goes international. And that one can be stored at normal fridge temperatures. So freezer temperatures at the moment, hopefully normal fridge temperatures soon. And basically, I think you know, for big rollout programs like in poorer countries, or you know, if it was being rolled out in Africa or Asia, these South America, whatever, these very cold temperatures will be very difficult. So fridge temperatures will be necessary, and it looks like they're doing that. Developing an alternative formulation that can be refrigerated, so that's what they're developing now. The freeze-dried form, so that's good. This is the Gamaleya Research Institute, part of the Ministry of Health in Russia, that's developed this vaccine. And it's a combination of 2 adenoviruses. Now, there's that one, Ad5 and Ad26. So there's 2 adenoviruses. And as we'll see, they give... I'd initially thought they'd put both adenovirus vectors in one shot of vaccine. They haven't. They're actually giving one type of adenovirus vector with the first shot and another type of adenovirus vector with the second shot to prevent people becoming immune to the adenovirus vector. Now, this has been done in other things, and it's a novel approach, and it does make quite a lot of sense, actually. And this kind of mix and match approach, well, it's not quite mix and match, this is done deliberately, but it's probably got some efficacy. So there's a new trial started in the UK at the moment, where they're looking at giving people the first dose of, say, the Oxford vaccine, and then as a second dose giving the Pfizer vaccine. So combining different types of vaccine because they've got slightly different immunological responses. And there's a trial just starting in the UK looking at that to see if it gives higher levels of efficacy. Now, we're not doing that in the UK at the moment. The guidelines at the moment say you should have the same vaccine for your first dose and your second dose, for your priming dose and your booster dose. But it's an interesting possibility that you could actually get increased immune response by giving different vaccines for the first and the second dose. And of course, that means if we do happen to need an extra booster dose at the end of a year, then in theory that would be so much the better as well. So interesting refinements on our approach to vaccination coming up here. So 11th of August, President Putin announced approval before the trials were done, not the way everyone else does it. That's what caused a lot of the controversy. They did not do it the normal way. So before the phase 3 trials had even started, it's not the way we normally do it. November, Russian government began offering Sputnik V within Russia, but there was widespread hesitancy, I think largely because of these two problems. But I think this paper basically answers the concerns that I had, and indeed the people of Russia had as well. Very reassuring to see it in the peer-reviewed literature. And we said this at the time. We said, look, if this vaccine's good, do the trials, put it in the peer-reviewed literature, let the whole world review it. And believe me, people all around the world are looking at this paper now with some well, they're astutely looking at this paper now. So, but this is the way science works. And if the feedback's not brilliant, the feedback's not brilliant. But it's peer-reviewed so far, so basically everything's back on track. Phase 3 trials carried out in Belarus, United Arab Emirates, Venezuela and India. But this paper we're looking at now, the data is only collected from the Russian sample. So it is very intermediate data. But that's fine, we're in an emergency situation. Now, Gamaleya Institute joined forces in December with AstraZeneca. Now this is unusual. Now, at the start of this program, at the start of the Russian program, there was some international agencies suggested that Russian sources had been hacking into vaccine programs from overseas. But now it's all legitimate, so there is complete cooperation, as I understand it, collaboration between the Gamaleya Institute and AstraZeneca, and they're hoping to synergize aspects of their program, which is exactly the way things should be done. Let's have the very best of the Russian science, let's have the very best of the Oxford AstraZeneca science, put them together and synthesize something even better. Delighted to see that this is happening. Now, the AstraZeneca Gamaleya combined vaccine started phase 1 trials on the 24th of December. So, you know, who knows? This might turn out to be the best vaccine in the world. We don't know yet, the phase 1. So it's just started. The phase 1 trials have just started, but they've come up between them with a novel new vaccine, which is now being assessed. So sounds good. Right, let's get on to the paper itself. So this is the paper here. Let me just show you it. There you go. Whole thing's there. Abstract, tables, full text, everything. Spent quite a bit of time looking at this last few hours and last night. All there for your perusal, completely free, public domain. So don't know if it's available in different languages or not. It may well be available in Russian, I'm not sure. Anyway, so it's there. There you go. Safety and efficacy of their vaccine. Right. Now, I'll tell you what some of this title stuff means now. So that is the actual text I've just looked at there. Click on it and you'll go straight to it. And this is another write-up in the Lancet describing this and sort of discussing it. Now, background. It's a heterologous recombinant adenovirus. Now, hetero, of course, means different. Now, the heterologous bit here, it doesn't mean that it's two different adenovirus vectors. What this means, actually, it's hetero because it contains genetic material from the adenovirus. Of course, it is an adenovirus. But it also contains synthesized genetic material that codes for the spike protein on the SARS coronavirus too. So it's kind of a transgenic organism, really. It's got a bit of adenovirus in it. It's got a bit of SARS coronavirus 2 genetics in it, and that makes it heterologous. That's what that term means. Not a common, we don't use that term very often in genetics, but that's what it means. So phase 2 results were good. Phase 2 results showed a good safety profile and induced a strong humoral, that's the antibodies, and cellular immune response. So the humoral response, the antibodies, very often, well, it is lasting quite well actually, but it might not last for very long, whereas the cellular response could well last for years and decades. Remember the memory cells, the memory T cells and the memory B cells. That's what we mean by the cellular response. Now, this is an interim analysis of the phase 3 data that this study is looking at. So presumably what they'll be doing is they'll be combining the data from Belarus, United Arab Emirates, Venezuela and India into a final paper. But this one is still good. Method. Randomised double-blind placebo-controlled phase 3 trial as you would expect. Data from 25 hospitals and polyclinics in Moscow. So it's actually Russian data at the moment. Would have been nice to see it from more countries. Would have been nice to see other areas in Russia. But you know the numbers are good, as we'll see. Participants 18 years old, and they were tested negative for the antigen and the antibodies, both IgG and IgM. So in other words, they hadn't been exposed to coronavirus before. And they were randomly at size 3 to 1. So 3 went into the vaccine group, 1 went into the placebo group. Different to the way it's been done in the UK, but nothing wrong with it. 2 shots 21 days apart. First dose is that one, second dose is that one. So adenovirus 26, recombinant adenovirus 26, recombinant adenovirus 5 for the first dose. So I haven't seen this, but I assume what they must have is they must have like 2 vials, and it's got dose 1 and dose 2 on it, because they're both different. So and this is the only one. This is the only vaccine doing this, as far as I am aware. So presumably they must have 2 vials. Maybe they're red and blue or something. That's the way I would do it. I'd have them different colours. But anyway, there must be 2 different ones for the first and the second dose. But both vectors carrying, so the vector is the adenovirus, carrying a gene for the full length of the SARS coronavirus 2 glycoprotein spike protein. So as we know, that is the bit on the end of the SARS coronavirus 2 virus, not drawn to scale, of course, that actually binds into the ACE receptors on the cells to cause the infection. So basically they've synthesized a bit of this, and that's what the adenovirus is taking into the body, and that will allow the body cells to make its own antigen. Just the same way as the very similar way to as the Oxford AstraZeneca vaccine works. Outcomes were recorded from 21 days after the outcome recorded 21 days after the second shot. Okay, that's fine. So they gave the second shot, waited 3 weeks, and saw what the results were. And we already know that the results are good. So let's look at them. So findings, data collected from the there we go. Data collected. Am I on? Yes. Data collected from the 7th of September to the 24th of November. Now, to be fair, not too long a time period, but we know there was an awful lot of there we go. We know there was an awful lot of cases in Russia at that time. An awful lot of infection in Russia at that time. So they probably wouldn't take too long to collect this data. 21,977, so 22,000 people initially joined the trial, randomly assigned to the vaccine or placebo group. They ended up with 16,501 in the vaccine group, placebo group 5,476. Now, the fact that these numbers are different doesn't matter too much, because the mathematics can take care of that. The number that actually got through to receiving 2 doses were 19,866. So they were included. And importantly, 2,144 participants older than 60 years, 1,611 in the vaccine group and 533 in the placebo group. And the whole study was stratified for age, of course. They collected the age information of everyone who was in the study, but a good number of older participants. Primary outcome analysis. Participants who included 2 shots at times of database lock. That's when the study was ended. Now, finally we get round to looking at the results. Sorry, a bit of technical issues today. I think we're there. Yes, right. So the vaccine group, they ended up with just under 15,000 people received the vaccine. And in that group, 16 confirmed COVID infections. So 16 of the 14,964 people got COVID-19. But after 21 days after the first dose, no cases of moderate or severe COVID-19 confirmed at least 21 days after the first dose. So 16 people, 0.1% got it. But if you waited 3 weeks after the first dose, no one got very sick. So very similar data to the Oxford vaccine. In the Oxford vaccine, no one got very sick after 22 days. I don't think there was any hospitalisations at all with the Oxford AstraZeneca vaccine after 22 days. And it was the same with the Sputnik V. Sputnik vaccine was the same. So after 21 days, people are kept out of hospital, which is absolutely marvellous news. That's what we want. But in terms of preventing the infection, so 16, that's 0.01%, got the infection after they'd had the vaccine. Now the placebo, they gave the same buffer. So the placebo was made up by the same company that made up the vaccine. And they had the same buffer, like the same solution that the active part of the virus, the vaccine was in. But of course, they left the active part of the vaccine out. So it was very similar, but didn't contain the active ingredient. And here, 62 out of 4,902 got COVID-19, which is 1.3%. So we see that 0.1% of the vaccinated group got it and 1.3% of the placebo group got it. But in the placebo group, 20 cases of moderate or severe COVID-19 confirmed at least 21 days after the first dose, which of course was the placebo. So 20 people got quite sick in the placebo group. No one got quite sick in the vaccine group. 0.1% of the vaccine group got the infection, although they were all mild. In the placebo group, 1.3% got the infection, 20 of which were severe. And you take those figures, and that comes up that this is 91.6% better. So the efficacy, this is preventing 91.6% of infections. Good number, good efficacy. And in older participants, it went up to 91.8% efficacy. Now, I don't think that was probably significantly different, but what it means is in older people over the age of 60, the response there looks slightly better. I think we can say the response in older adults is as good. So also very promising. So that was the epidemiological data. What about the lab data? Did that coincide? Did that concur? Yes, it did. So interim immunogenicity, how it generates immunity in the body. Vaccine group, receptor binding domain specific immunoglobulin detected in 98% of samples. So the seroconversion rate actually turned out to be 98.25%. So the vast majority of people making antibodies, the IgG antibodies last for a good few months, of course. So that's good. So it's good. They were seroconverting. High rates of interferon gamma were also produced. Now, interferon gamma is a measure for cellular immunity. So we have all these immune cells. So, for example, the interferon gamma is secreted by the T cells that we know about, the T lymphocytes, the T cytotoxic lymphocytes, the B lymphocytes, and the natural killer cells. So these immune cells, this cellular response, which of course is the longer lasting response, that was present because there were high levels of interferon gamma there. And we know that when you get these activated T cells, the subpopulation of those becomes memory T cells, and they can hang around for a long period of time. So in other words, we were getting good vaccine response, good antibody response, and good longer term cellular immunity response. So all checking out. And there were significantly higher levels of interferon upon secretion. So basically what they did is they took some blood and reintroduced the antigen, and they got lots of this interferon. But it was significantly higher. So the vaccinated group had significantly higher levels of interferon compared to the placebo group. Now, the placebo group, the immunoglobulins there were detected in 17, that's 15% of the sample, as opposed to 98% in the vaccine group. So how did this placebo group get it? Well, that proportion must have been exposed to the virus through natural exposure. But again, big difference, significant difference between the two groups. So 98% of the people with the vaccine are making the antibodies. That is the key take-home figure. Seroconversion rate was 15% in the placebo group, and that's of course related to 98.25% seroconversion in the vaccine group. So that's the difference between the two. Significantly better response in the vaccine group. And the probability of that happening was not 1 in 10, it was not 1 in 100, it was not 1 in 1,000, it was 1 in 10,000. That result would have arisen by chance. P-value is 0.0001. So efficacy looking good. Now in this study there was no evidence that, as we have for the Oxford vaccine, there was no evidence that it's preventing person-to-person transmission. But that doesn't mean to say that it wasn't. It may well have been, and I strongly suspect it was. It's just that the evidence was not there for that. But I would be surprised with this level of efficacy if it wasn't preventing contagion by reducing the viral load so dramatically. There were no viruses there to pass from person to person. I think the reduction after one dose of the Oxford vaccine was 67% yesterday, wasn't it? So I would expect this to be the same, higher after 2 doses. But having said that, we simply don't know. The data's not there. Adverse events, grade 1, so minor things, sore arms, sniffles, headaches, minor transient effects. So out of the total adverse events, 7,966 adverse events for everyone, the placebo group and the experimental group. 7,485 of those were minor. So that means there were some serious adverse events. Now in the vaccine group, there was 45 adverse events out of 16,427 people who received the vaccine, and in the placebo group there was 23 out of 5,435 people who received the placebo. So the adverse events in the vaccine group were 0.3%, and the adverse events in the placebo group were 0.4%. In other words, they were basically the same in both groups. And all of the adverse events were looked at in detail, and none were considered to be associated with the vaccine. So safety profile looking good, as with the Oxford and the other vaccines that have already gained approval. 4 deaths were reported during the study, which we would expect because we're dealing with nearly 20,000 people, 16, 17,000 people, whatever it was. Vaccine group, there was 3 deaths, less than 0.01%. 3 out of 16,427. Placebo group, there was 1 death out of 5,435. So we see about the same level of deaths in the vaccine and the placebo group. It's just so great to have 2 groups to compare, isn't it? You just know what you're doing all the time. But the deaths, again, were reviewed, of course, by expert committee, and none were considered to relate to the vaccine. Interpretation, this is directly from the paper. This interim analysis of phase 3 trial of the Gam-Covid-Vac, what we've always called the Sputnik V, the Sputnik vaccine, showed 91.6% efficacy against COVID-19 and was well tolerated in a large cohort. So the question is in my mind now, will this be substantiated when we get the international data? It would be surprising if it wasn't. It should be given, but you can't extrapolate too much from country to country, but I would expect it to be. We're looking forward to the final phase 3 data. Is this enough for regulators to give approval on? Well, yes, it is. Hungary, for example, I believe has already approved it. And Serbia has. Of course, it's been used in Russia. So Russia, Hungary, Serbia that I know of at the moment. Oh, the other thing I don't know yet is what the Russian manufacturing capacity is. Will they be able to ramp this up massively to supply all the countries round about, for example, and contribute towards the COVAX programme? Don't know the answer to that. We'll check that out. I'm not sure it's massive at the moment, but I strongly suspect the Russians are trying to ramp it up big time. So basically, as long as the final phase 3 data checks out with the international data, it looks like for the Sputnik vaccine the only way is up. It's looking good, and my view is now that given the data we have, this can supply, this all will provide a significant component of the global fight against this pandemic. So that is where the evidence is at the moment for that. So it is very, very good news. Good news from the Oxford vaccine yesterday, more good news from the Sputnik vaccine today. Let's close with a few pictures. Now, this is Amber from Alabama. And you might think Amber's looking a bit pleased with herself, and she is, and rightly so, because she's just had the Moderna vaccine. So great news, Amber. Do keep, as you know, behaving as you have been for some time yet. Now I've put this picture on again because I think it's a great picture from Debbie. We did see it last week. But it's just such an expressive picture that she's keeping people away with her red pole. Social distancing. As long as the ventilation in the room is also good. Great picture. Just explains it so well, doesn't it? And this is Dee, who watches while she does her artwork. So thank you for watching, Dee. That is good news. So, the end of this pandemic now looks more attainable as more vaccines come online, and the vaccine news basically is just consistently good. You know, we've got several vaccines coming along. We've got the Johnson & Johnson, Janssen one, that other one we looked at the other day. What was that called? Anyway, there's a few coming on, and vaccinations will be rolled out massively. Rich countries are going to be vaccinated basically this year, and let's just hope we can roll this vaccine out to everyone, or these vaccines out to everyone who needs them. Oh, just one other thing on the vaccine front. There were some worries, of course, about the new mutations, which are a concern. But we know that the vaccines we have at the moment are quite good against the UK mutation, slightly less so against the South African mutation, we believe. But the Oxford team at the moment is working on a second generation vaccine that should be ready in the autumn, that's presumably more specific against the B117 UK variant and potentially the South Africa variant as well. So the vaccine manufacturing teams are capable that they can adapt to new mutations that are coming along and will continue to come along, because we know this is a mutating virus. These mutations are going to come along until we are able to eradicate it, and I still believe we're going to be able to eradicate it. I've been saying this for about a year now, and I believe that this good vaccine news is another reinforcement of that argument, that we will be able to eradicate this virus. Others will come along, but hopefully we'll have learned from this experience and are in a much better position to deal with the other viruses that will come along. Okay, that is us for today. It's another good news video, so I'm just so grateful that you've stayed till the end, those of you that have, and thank you for watching.